Hi everyone, it is great to be back here. Thank you very much for inviting me again, Felix. I really appreciate it. So today we're going to talk about a lot of topics, some of which are dry in bioinformatics and some of which are wet. And hopefully some of these are going to jog a memory in your mind on the processes we use to figure out what a new sequence is capable of doing. So we're going to try to walk through several methods for that purpose. Oh my goodness, well, just come back around here. Is the screen actually responding? Yeah, that's good. All right. Let's try that. There we go. Perfect. All right. Now, there are quite a few little pieces to the talk, so I'm going to try to focus around three big questions that we could ask of any new gene. So we're going to start with, have I seen a sequence similar to this one? And that's going to have us talk about BLAST and InterPro and ProteinOrtho. All of these fall in the category of things that we would call sequence homology. Can we find homology between a sequence and another one that hopefully has been characterized a little better? Next, what genes have been expressed similarly to this one? We're going to talk about the mechanisms by which we develop our gene-to-gene relationships to understand where these gene networks come from. For that, we're going to talk about something that occasionally gets called functional genomics. I've always sort of disliked that term because it kind of oversteps the fact that transcriptomics is in many ways a field of its own. But along the way, we'll spend a lot of time talking about gene expression correlation. How do we determine sets of genes that go up and down together according to the same stimuli? Finally, does my gene product collaborate? I mean, you can guess which professors are the good ones to work with because they're teammates for everybody, right? In the same way, genes that have relationships with lots of others tend to be those that have very interesting dynamics in our systems. So we'll spend a fair bit of time talking about protein-protein interactions, or PPI networks. It's fine to talk about gene expression networks, but how the proteins interact can often be quite interesting and not really visible when we look at it from a transcript-only perspective. And for that, we'll talk about co-localization, the yeast-2 hybrid system, and co-immunoprecipitation. And hopefully we all learned something new today. All right, so I'm going to start with the fact that a lot of times the new sequence data we have is coming out of either gene sequencing or transcript sequencing. These days you can do RNA-seq on pretty much any species, even if you don't have a gene reference, a genomic reference to go against. So we might talk about some basics here, that one of the things that you might assume is that starting with transcript, the gene level sequence is obviously the way to go. The reality, however, is that if you're looking for distant evolutionary relationships, you're often much better off to be working with a polypeptide or protein sequence instead. So I would note that given a particular transcript sequence, there are 6 ways to translate it. Don't forget that we can do work in reading frame 1, 2, or 3, and the complement 1, 2, 4, 5, and 6 in that case. So a single transcript sequence that you feed in may be translated any of 6 ways. I often find that the tools produced by MBOSS are very handy for this. MBOSS has a whole pile of web servers that I think are very well known, but you can also run MBOSS on your own computer. This is very useful if what you have is a RNA-seq transcriptome that you've assembled right out of RNA reads, right, or out of cDNA reads anyway. So in a case like that, you may have thousands of transcripts and you're trying to figure out what subset of them relate to a particular process. If you're in a position like that, being able to run things in batch makes a very big difference. You can run MBOSS right on your own desktop. And MBOSS includes tools like transseq, which will let you generate a 6-frame translation from a whole FASTA full of transcripts. And it also includes a very helpful tool called checktrans. How do you make sense of the data that come back from something like an assembled RNA-seq transcriptome? Maybe you've got 100,000 transcripts that got assembled from an RNA-seq experiment. How do you figure out which reading frame is appropriate for all of those, right? So frequently, one of the criteria we use is a pretty simple one. Having translated a transcript to 6 different polypeptide reading frames, we would then ask which one gives us a nice long open reading frame. If it's all chopped up into tiny pieces, that's not likely to be the real reading frame. So the check-trans software can let you apply a threshold to say, I'm only interested in the translations that are at least 100 amino acids in length. So these software tools are right there. Many of the tools we would use to evaluate those sequences tend to be much, much faster when given protein sequences than when given transcripts. All right. So at the left at the top, I've given a sequence that we've assembled out of hyena. I was trying to explain to the honor students a couple years back why we need to study hyena. And I started with some questions about immunology, right? If a hyena eats something that's sick, why doesn't the hyena get sick? And I didn't feel like I was really connecting with them until I finally said, look, the world is running out of fuzzy animals that have been sequenced, right? That haven't been sequenced. Let's do this. They got it. All right, so this is a sequence that we assembled out of RNA-seq. I wanted to note that I've already cheated a little bit in putting this together. I've got an arrow pointing up there to a very small bit of squibble of text, which is a series of ends. So I've actually grabbed, I had a case where my transcript was split to two different assemblies. So I've stitched it back together just to make it a little easier. So I've written in that 6 nucleotides are missing there, and I filled them in with ends. Now at the right, I've applied the software, the TransSeq software from MBOS, and it gave me this back as one of the translations. So I noted that we had non-canonical nucleotides over there marked as N. You can see that over here, those have become X's in this case. So we don't know which amino acids go there. But we also have these stars in here. Does everyone remember what a star might represent in a polypeptide sequence? Stop codon. Got it in 1. Brilliant. Okay. He's reading ahead. You have to ask, if you attend Journal Club and you actually read the article in advance, does that count as cheating? It doesn't. It doesn't. Okay, so here we have a translation, the open reading frame pulled from this. And if we read from stop to stop, we have this series of amino acids that we believe are going to represent the sequence for this particular gene. Now, how do I figure out what this is? You can see at the bottom that this is a Trinity gene assembly. So Trinity always puts its name in front of the gene name. In this case, it's DN3H2H9. Does that represent anything biological? It does not. It's mostly about what address number that came from. Out of all of the genes that were assembled, which number did this one get? So what can we do with this? I'm going to start with InterPro. A lot of people interact with InterPro through the website, and that's great if you're going one protein at a time. If, however, you are dealing with thousands of proteins at a time, you would better have a system that can let you run this in batch. So helpfully, InterPro has a version that you can download and run on a Linux computer. It is about 50 gigs in size to download. It is a big piece of software. But it's big because it has all of these different fingerprints in it to explain what little bits of sequence relate to what known functions. So InterPro is a really handy first pass on a novel transcriptome to figure out which functionality is found in which of your transcripts. In this case, I've passed it the sequence that we were looking at from stop to stop in the prior slide. And you can see that InterPro is now going to show on the x-axis where particular features may be found within this polypeptide sequence. We have a variety of different kinds of signatures that are going to match. Family, homologous superfamily, unintegrated in this case, which means it's a new signature that hasn't been sort of migrated into the main body of InterPro yet. And then some predictions down here at the bottom. Now I'm going to give away the story in the very first part of this slide. These top signatures that we all match are family hits to interferon gamma. You see, I have fallen in with immunologists, and that's the people who funded this study. They care about the immunology of hyena, which means that their pressing question was, what is the sequence for the interferon gamma in hyena? They've been trying for years to clone this bloody thing, and now here we've got this massive RNA-seq data set, and in one swoop we can say, this is what the sequence for it is. So we were very grateful that we got really strong family hits then to the interferon gamma proteins. At the larger level, we might ask what larger family this can fit into, and the superfamilies help us address that. These are 4 helical cytokines, so it's a class of cytokines that include the interferon gammas. And Down here, under unintegrated, what seems like kind of a miscellaneous category, we find that we have a hit to gene 3D. Now, when I get to the next slide, we're going to explore just what kind of information we can get from that. But down here, I don't want to forget that we have all of these little marks at the lower left. If you have a whole bunch of very short signatures that hit on the N-terminus of a protein, do you have any suggestions about what those might be? I think I heard it. Signal peptides, very good. Right, well, whenever you send a shipment on dry ice to another institution in South Africa, you put an address label on it, right, to explain the shipping and handling of that package. In the same way, these proteins come with these very handy labels on the front to say, where am I supposed to go once I'm assembled? That's very handy. So the signal peptide regions have all been labeled by different pieces of the InterPro signature. Now let's come back to this unintegrated gene 3D piece. If I click around a little bit on that gene 3D hit, I see that not only are there sequence matches to different parts of the structure, we have structural matches, and the structural match in this case covers a very long swath of that amino acid sequence. So that's brilliant. In this case, it points to a particular CAF family. These CAF families are associated with particular structures known to be conserved across species. So we have a great many sequences seen across a large number of species that all relate to this same fold. That's brilliant. I mean, you're not going to get a whole lot closer to really good to really good functional information than a really decent structure. So we're grateful to have that. That's lovely. And it helpfully comes with the sequence alignment. Up at the top is a sequence logo, which says essentially how dominant is each letter at each position. Very useful for understanding which parts of the sequence are modifiable and which ones will break it irretrievably if you change them. And we even get this possum. Does everyone remember possums? PSSM? Now, in the United States, a possum is that animal that makes the noise in your trash bin at night. Right, that's not what we're talking about. This is a position-specific scoring matrix for this sequence motif. So we can see that a whole bunch of different examples of proteins of this type have all been stacked together to show us what kind of sequence heterogeneity may be found within these sites. So great information in these. You don't have to just look at the overall picture. You can drill into it and get information about why this sequence motif was a match. All right, let us move ahead. Now, I think a lot of people in this world assume that bioinformatics is all about BLAST. BLAST got published a long time ago. We haven't stopped there. There's a lot going on, but BLAST is still useful. So in this case, I have run NCBI BLAST over at the European Bioinformatics Institute. I don't know why. It's simply a lot easier to use this tool in their web interface than most anybody else's. When I'm trying to figure out where to do my sequence alignment, it's almost always the case that EBI UP has got me covered. So here we go. That commercial is now over. So we have run a search now against all of the UniProt knowledge base. Everyone remembers, hopefully, that UniProt consists of SwissProt, which is all the manually curated protein sequences, and Tremble, which is all the automated stuff that hasn't been human curated yet. And I gotta say, the computers are always adding to the size of the stuff that hasn't been human curated yet, no matter how hard those curators work. What we have are sequence matches to show us within the sequence we submitted where the matching part of sequence was for what appeared in UniProt knowledge base. In this case, I've supplied a messenger RNA sequence, that was the one at the left, instead of the protein sequence, because I thought it might be decent to do a search with the transcript rather than the protein. In this case, BLASTX was making a comparison then between amino acid sequences on the one hand and a transcript sequence is what I handed it. If I had done a protein versus protein one, the result would probably have been much the same, except it would have used BLASTP instead of BLASTX. Minor difference. These E values that come back are our way of evaluating how random a match this was. What is a terrible E score? If I get back a best hit from a search and it has an E value of 1, am I excited or am I sad? Sad. Okay. What that means is this result scored just as well as you would have expected by random chance if you'd had only random matching throughout this database. A 1 is terrible. So what we're really looking for are these vanishingly small numbers as e-value scores. And in this case, all of these are brilliant hits. Now, I realize that it's far too small to see, but I'm going to highlight that because I did my search against UniProt knowledge base, each of these has information in the tail end of the accession that specifies what species this came from. So just at a glance, hyena is related to other species, but what's its closest sequenced relative? Blast has our answer. Up at the top of the list, we have a whole bunch of phelcas, and that is a house cat. Very short story. The #1 center on Earth for sequencing cats is the University of Missouri, the place where I spent my very first internship. I'm from Missouri, way back in the day. So I have to say, the first cat ever sequenced was named Cinnamon, and he was a Missourian. Yeah, all right. So we see then that the house cat is #1 on this list, the cheetah is #2, the lynx #3, the tiger #3. Hyenas are not dogs. They're cats. Who knew? So that's brilliant. We can use InterPro to find these domains and motifs that we can match to. We can use BLAST to match local alignments elsewhere. But in this case, we want to do a really large-scale analysis. Does everyone understand the difference between paralogs and orthologs? Some yeses, some nos? Okay. Over our evolutionary history, our bodies have evolved duplicates of certain parts of our genome. Now, the usual circumstance when you duplicate part of your genome is that the duplicate goes away. You don't need twice as many genes producing copies of that thing. But on occasion, we find that gene duplicates functionally diverge from each other. In fact, we find that genes that were once soluble enzymes end up doing things like refracting light well, that the crystallins in your eye have diverged so much from the original enzymes that they now play a more or less structural role in the body. So this divergence is called paralogy. It is also the case that we share genes with other species. And as soon as you share a sequence homology across a species boundary, you call it an ortholog. So if you're talking about the same gene across species boundaries, you're talking about orthology. If you have assembled an RNA transcriptome from a bunch of RNA-seq experiments and you know nothing else about this species, one of the natural questions you're going to ask is how the transcripts you've produced from this species relate to that same gene in another species, or at least the closest, our best guess as to which is the current one. For that, you're going to need some strategy by which you can perform alignments between all of these thousands, tens of thousands of sequences you've derived to the transcriptome of known species. So ProteinOrtho is a tool that we found to be really, really useful for this purpose. The original releases of ProteinOrtho actually ran BLAST on your computer itself against databases you had assembled. More recently, they've switched to some software called Diamond. Diamond is an extraordinarily fast protein-to-protein matcher. So it is, just to give you a notion, I thought that I was going to keep my house warm for a couple weeks this winter when I handed the software many plant proteomes, whole collections of plant proteins from 6 or 7 species. I thought it's going to take it forever to compare all of these protein sequences to all of those, and to all of those, and to all of those. It was done before I went to bed that night. Right, so, I mean, I have a great home PC. But just the same, a lot of the sequence homology stuff has gotten a lot faster, and we have some really great strategies for accelerating them. ProteinOrtho will handle this process of comparing all of these proteins to all of those proteins to all of those other proteins for, say, 10 species at a time. It's really useful when you're trying to recognize orthologs that may be present in some species, but not present in others. So I would really recommend everyone take a shot of that if you're trying to achieve this on your own projects. Whew! Well, that was quite a lot of sequence homology all in one quick go. So let's instead talk about this functional genomics problem, a phrase that I once swore I would never say. Gene expression. How can we evaluate the functional significance of a novel protein sequence or a novel gene sequence based on its expression in different circumstances? So let's imagine that we have a particular species that's now been evaluated. We've done maybe RNA-seq in 100 different experiments or more that show different kinds of stimulations of these. We've got some examples of the species with cells growing in exponential phase and some in lag. Some we've tortured them with a very unfamiliar carbon source, whatever. And we can do this, of course, with animals as well. Preferably we don't do it with patients, but So we have all of these different kinds of ways that we've, different growth conditions in which these cells have been tested. And in each case, we have a gene expression metric to evaluate the level at which each transcript is being produced. So we can think of this then. Ideally, you did this in a microarray, right? Because then your data capture costs are a lot lower. So our microarrays have been considered across all of these different conditions, and in each case, the genes have been measured. The same set of genes has been measured. We now want to understand which genes come up and down by the same subsets of stimuli. Now, there's this rumor that there are housekeeping genes, that there are genes that are always on and are invariant in level no matter what's going on to the cell. This is a rumor, it is not truth. So if you were planning on using housekeeping genes to normalize away this behavior, just know that there's a bioinformaticist ready to sneer. Okay, right, so we have these microarrays now, and these gene measurements across all of these different stimuli. We can decompose this as a correlative or correlational prospect, that we can say genes that are up together and down together are similarly are participating in similar functions, ideally. Now we could do it by a correlation process. In this case, they're showing it via ICA decomposition. So I want to show you that, in effect, this process is going to untangle these gene and microarray relationships by instead relating the different conditions to modules. You can think of this as kind of a PCA thing, right? That we have all these different genes and they have different group memberships. So we're decomposing them into those groups in which they're functioning. And from that, we get an association of genes to those groups. So this decompositional process is saying genes that go up and down together, I'm going to group together in modules, where I have a smaller number of modules to represent the whole system that can then be mapped to genes. That is a bit to swallow, but that is a process that has become a standard way by which we discern the transcriptional relationships of different genes. I thought that the way they put it in this paper was very good. I apologize, I'm here citing a preprint. You will forgive me, I hope. They just had the right words and the right figure. Gene expression data can be used to define groups of genes that show similar patterns of expression or covariation across multiple conditions. This is one of our biggest tools in relating novel genes to those that have been more completely annotated. There are many, many, many ways that you can work from a set of microarray measurements to these sets of modules. Quite a variety to work with. We've got in here regression and mutual information. Has anyone heard of mutual information? Do we have any computer scientists in the room? Okay, well, a lot of people who work in information theory think of mutual information as kind of how many bits separate the behavior of these things. So if they have a common behavior across all of these conditions, they have a high degree of mutual information. So mutual information is a route, correlation is a way, Bayesian networks are popular. There are some approaches even that make use of machine learning. For example, random forests. I swear every time we say machine learning in my group, everyone has to take a drink, but I've only got my bottle of water here. So machine learning is a very, very big deal in bioinformatics, and it seems to be poking its nose in just everywhere. So in this case, machine learning has found its way into discerning which genes are going up and down together in expression. I've honored the tradition. On we go. There was a really interesting paper back in 2012. I believe this was from a group called Dream5. They were making a competition to say, we're going to give all of you the same set of microarrays, and we want you to use your published technique, your all-star, to infer sets of networks. And then we're going to see if people given the same data agree or disagree about which genes are most similar to each other. Very straightforward exercise. So you can see each of these circles represents another one of these teams participating in the Dream5 competition. What they found was that if you look at an individual lab's performance on these, on what was actually known and on their consistency with what others found, some labs scored brilliantly by themselves. In the mean, most groups using a single method for this clustering approach did rather poorly compared to the overall assessment strategy. If, however, we were to combine information from multiple approaches for evaluating these gene expression data, things got rather better. That it's easy to play these different methods off of each other. So instead of the Bayesians saying only the Bayesian approach could plausibly be used to make any reasonable sense here, it makes a little sense to include a Bayesian in your analysis and to include a computer scientist in your analysis, maybe include a random forest. That using multiple approaches to analyze these data is much more effective at getting information out than any one method alone. Which suggests to me that there's a lot of room for improvement yet in these methods. So what comes out of that? I think most of us have encountered diagrams like these in a paper. Frequently, they're showing it such a huge number of genes at such a scale that it looks like a hairball, frankly. So here we're looking at one that's a little simpler. This is just from Staph aureus, so a relatively compact one. This is a consensus approach using a community network that's derived from all of those different methods, analyzing the same input data. So at this scale, it's still quite noisy. It's quite hard to interpret that, I think. But as we look at a tiny little leaf on that diagram, we start seeing something more like this. Now, I could talk at this point about the hierarchical modular nature of genetic networks. We could talk about scale-free networks or how they're small world. But I would just point out that in this case, what we see are a few transcription factors, in this case, which have a shared set of targets that have these related functions to work with. So this has been really valuable in that they've created kind of a directional network, that these transcription factors are driving this joint process together. That was published in 2012. There have been multiple rounds now published of these studies. Now, I'm a proteomics guy, and I've been working in that field a really long time, and I have to say, a lot of people feel that functional genomics is all about gene expression, and by that they mean transcription. You might think that if you know the messenger RNA level for a gene in a cell, that's all you really need to know. Of course, you know everything you need to know about how much protein is there. But I submit to you that the degradation processes and the generation processes for messenger RNA are rather different than what they are for protein. It may be that the amount of protein has very little relationship to how much of the transcript is present in the cell. So I would point you to this paper that caused us any amount of joy back in 2003 from Gami et al. The group created a tagging strategy that allowed them to quantify each protein individually and to measure each messenger RNA intensity separately. In this case, they were using microarrays, but we'll forgive them, it was 2003, right? So what you might expect is that if there's a direct relationship, a linear relationship, that if there's more messenger RNA than low, there shall be more protein, that what you're going to see is a big line here. This is not a line. What we see is an awful lot of scatter up and down, which reminds us that protein degradation and generation processes are regulated differently than are transcription and destruction of messenger RNA. So having this information is grand. It reminds us that if you want to study protein levels, you're going to need to study protein. Quantitative technologies in proteomics have gotten a lot better since I entered this field. I have to say that things were a bit wild and woolly in the late 1990s, for example. But today, there are a lot of technologies out there to enable us to quantify proteins at a very broad scale, to quantify 2,000 proteins in any of 50 or 500 samples even. So that's gotten much better. A Spearman rank correlation of 0.57 is not a story about causation, direct and complete causation. We haven't captured the processes that are destroying proteins, the processes of protein turnover. We're not capturing the processes of messenger RNA destruction. All right. So having spent a little time talking about sequence homology, having spent a little time talking about functional genomics, I figured it's time to start talking a little more closely about where our proteins are and how they are interacting with each other. This is kind of a challenging area to work in. I realized just how little I had retained about this from some of the really bright people. Barbara Trask was one of my teachers back in the University of Washington Med School on this subject. And I really feel like I need to return to those Med Sub days. Here we're looking at a paper from 2011, and it had a really useful subdivision of this idea of co-localization. I think most folks remember that red and green make yellow. That's exactly what we learned in elementary school, right? Not quite. When we have a green light and we mix it with a red light together, superimposed on each other, they should give us yellow light. So if you have a particular gene product, in this case, these are tagging proteins that are associated with endosomes, you would expect that a yellow light over here represents co-location, that we have the two different proteins showing up in the same place. These take different forms, though. Co-localization kind of spills over to cover all three of these concepts, and they really have different strengths and weaknesses and different capabilities from this type of technology. So we start with co-occurrence. Co-occurrence. If two different proteins are both expressed in the same kinds of subcellular structure, maybe endosomes in this case, then we can say that they co-occur. It may be that what we want is a more quantitative measurement. We want to say that where there's a lot of protein A, there's a lot of protein B. Where there's very little of protein A, there's very little of protein B. Co-occurrence doesn't address that. Co-occurrence just says where A is, so shall B. It's kind of a Ruthian thing to say, isn't it? Anyway, sorry, Sunday school lessons occasionally come back to mind. All right, correlation. So in correlation, we're trying to establish that where there's very little of a thing, there's very little of the other thing. Where there's a lot of a thing, there's a lot of the other thing. You expect, in fact, to find a relationship like this, right? This is an example of that correlative approach. Now, what if instead, in your overlay, you see a whole bunch of green dots or a whole bunch of red dots? These reflect proteins that are expressed differentially, that the red tagged protein is present a lot at that lower right-hand corner, and that right next to it, in the area next to it, you have a bunch of the green protein, but not a lot of the red protein. So these are standing out from this overall trend that we see in this case. One other thing to say about fluorescence microscopy like this, and that is about interaction. To say that two proteins show up in the same place and show up in correlated levels is all fine and good. But that is not to say that protein A interacts with protein B. Interaction is a much tougher criterion to reach. For that, you might need FRET. Do people remember FRET? Barbara Trask had so much to say about FRET. She was really fascinating. So FRET is a case where you have a fluorescent resonance where two dyes are interacting with each other, which requires a much closer touch than simply being in the same metric yard of space, basically. Metric meter, same cubic millimeter. So, right. Ideally, if you're looking for these interactions, you're able to show it through electron microscopy, right? If you can show that these two proteins are interacting at that level, then maybe you have a leg to stand on. But those are not within range of individually dyed proteins. I would also point out that there's a lot to be known about how to do proper image processing in this context. In this case, I wanted to bring up the importance of regions of interest. I note that in figure E over here, we have a rather greenish blob down at the lower left that's been boxed separately than the two cells next to it. And what we see is that the relationship between the red and green dyes in in the lower left one, is quite different than what you see in the two cells joining it. So if you were to look at this, as an overall thing, you're going to see that there are multiple patterns to be found within this image, multiple relationships of red to green. And if you set the region, I would start by saying that you want to be able to isolate the cells from everything behind them. That's what we call a segmentation process, or pulling the positive forward and pushing the negative back, essentially. Specifying that this image needs to be cut up into subfields, this one representing cell 1, this one representing cell 2, and this one representing cell 3, is going to give you very different kinds of information than if you try to examine try to process the entire field of view at once. Okay. Protein-protein interactions get misunderstood quite a lot. There's definitely a propensity in people who do proteomics for protein-protein interaction to try to claim the very largest number of interactions that could possibly be explained by a data set. It's a little dangerous. I felt like this was a really useful limitation on PPI. The physical contact considered in protein-protein interactions should be specific, not just all proteins that bump into each other by chance. It should also exclude interactions that a protein experiences when it is being made, folded, quality checked, or degraded. Otherwise, think about what a crazy situation you have in trying to explain the protein-protein interactions of ribosomal proteins. They touch everything. So obviously that's not a very useful thing. Chaperonins, they touch a lot of things. Proteins that, proteins that touch ubiquitin ligases, right? These are all examples of proteins involved in the management of other proteins. So claiming specific protein-protein interactions there is a little bit more limiting. So this was from their 2010 article. I thought it was a great definition to work with. This video and these slides will be available from YouTube, so I will include a link to the PDF there, so you can just re-download that if you need it. Back when I was in graduate school, the yeast-2 hybrid system was the bee's knees. Everyone was so excited about this. Having the ability to say that these gene products interact is fantastic. So now if we're able to create engineer a variant of the cell line in which the gene X has a binding domain attached to it, is great. Now I need another strain of this yeast in which this protein has been engineered with an activation domain on it. So if I am able to put these haploid strains together to create a diploid strain, if X and Y interact, then the binding domain and the activation domain are close together. And if they're close together, they can cause our reporter gene to produce whatever it's doing. So that's brilliant. This gave us the ability, for the first time really, to create massive libraries of these yeast strains that could then be combined to ask, does this gene interact with this other gene product? That's a brilliant thing. In effect, this is a bit of a false positive machine. Yeast-2 hybrid is really, really powerful, but it tends to over-report a little bit, that even incidental interactions between, in this case, protein X and Y, tended to lend themselves to this activation process. So we had to be very careful in how we interpreted yeast-2 hybrid data. They were a little prone to false positive interactions between things. For the last couple decades at least, there's been quite a lot of interest in using co-immunoprecipitation as a strategy for very sensitively probing, does protein X interact with protein Y, or a whole family of things. So where yeast-2 hybrid was a binary system, does this gene and this gene product interact, we're now able to pull out a bait, pull out a bait protein and see which prey proteins come along with it. And in the early days of this, at least, people were very prone to saying that if you saw protein P, when you pulled out protein B, that the two interacted, of course. There are lots of reasons why that's not safe, though. Alexei Nesvishky, a friend of mine at the University of Michigan, has done a lot of work on this with an algorithm called SAINT. You might also see his papers based on what he calls the crapome, reflecting the false positive interactions that can occur in this case. You would like to believe that you've used your support coupled with an antibody to pull out a tag, and that tag was engineered onto the protein you were going to measure. From that, the only proteins other than the tagged antibody and the bait protein should be the prey proteins. What Alexei has helped us to understand is that in fact there are a great many contaminant proteins. Some of them associate with the surface, the support. Some of them associate with the antibody. Some of them associate with the tag. Some of them associate with the bait. What happens if you have a prey protein that has its own prey proteins? Is that a direct interaction? Not necessarily, but will the proteomics data tell you that that is an additional interaction, a secondary interaction? Not necessarily. It'll simply give you a list of what's there. So Alexei's software is some of the best out there, I think, for being able to tangle through lots of data from these co-immunoprecipitations and tell you which ones are the most specific interactions, which are simply sticky proteins known to stick around to lots of things. Over time, people have gotten more and more subtle about how they go after bait proteins. They found themselves thinking, if I can grab this bait protein with this one tag, I could make my result more specific if I do a double affinity, a tandem affinity purification, a TAP tag. Using a pair of these affinity approaches often gives us a much cleaner set of data to work from, fewer contaminant proteins that we have to worry about. Now, you might imagine that if you go through a tandem affinity process, you might disrupt a real interaction and lose one of the prey proteins that you actually want to keep. But tandem affinities become quite popular. It is also, however, kind of a flavor of the month club, in that we frequently find that there's a new paper every month announcing some combination of affinity tags for this purpose. So I like the fact that Yifeng Li put together this paper in 2010 to say which ones are most popular out of this dessert buffet, basically, that's being presented in the proteomics literature. So hist tags are quite popular. So if you find a whole bunch of histidine residues used as a tag, you can pull those out with an IMEx column. So immobilized metal helps us to grab out the hist tag, and thus you can pull along proteins in any dot to it that you have there. The flag tag is a very popular one as well. That's shown here in the orange sector at the right. You can grab that out with a monoclonal antibody. And protein A is a very popular one. This is, I believe, a bacterial protein that has a very strong interaction with immunoglobulins. So we have a variety of strategies that we use to lift proteins out of this mix. And hopefully, the purification processes are not disrupting real interactions and allowing us to have a cleaner data set to work with. I have just one more, a couple more notes actually before I want to go. So let's talk first about the maturity of pathway and network information in current databases. Isn't it possible that we could just look at the sequences, de novo fold them up, and deduce which proteins interact with which proteins? It's kind of a beautiful dream, isn't it? Well, some protein structures that are pretty easy to fold together. If you have a relatively short protein sequence and are trying to figure out how it's going to fold, you'll have better success than if you have one that's 1,000 amino acids in length. But we do have a fair amount of information now drawn from things like the protein data bank to help us assess whether a known structure gets us close to being able to say, this and this interact on this face. Localized regions on protein surfaces are conserved among structural neighbors, and they participate in these protein-protein interactions. We have evidence that helps us to trace which particular amino acid side chains are forming these interactions between a pair of proteins. That's a great start. As long as structural information is available for a given pair of proteins, hopefully when you read that you thought to yourself, well, now that's kind of the kicker, isn't it? Right now, Protein Databank has just over 160,000 protein structures. That's pretty cool. Many of them are redundant of each other, of course, that they are the same protein sequence appears in many PDB structures. But we don't have anything like a comprehensive structural identification of all human proteins, for example. And I don't believe that's going to happen in my lifetime, frankly. So this information is still lacking. But when you have it, the set of template complexes available in the current structured databases can be used to generate coarse-grained models of protein-protein interactions. This is kind of a cup half full, cup half empty thing. The availability of structures is the linchpin upon which we base our ability to discern protein-protein interfaces from a de novo point of view. Right now, the amount of information available in the databases makes it infeasible for us to find these interactions just de novo. You can't just plug in a pair of sequences and come up with a realistic appraisal of how they dock together. That said, protein-protein interaction databases have gotten really big and really complex. In this case, I am pulling together pathway databases and network databases, 3 of each. If you go to pathguide.org, you can see that there's a huge number of these relationship databases available to us. So I've just grabbed out 6, some of which you've probably heard of and some of which you probably haven't. Gene Ontology, I think everybody's heard of Go at this point. KEGG is a really useful pathway database if you're trying to understand all the different interactions that lead to a particular metabolite being created. Wiki Pathways is a publicly editable one, very valuable. String, IMEx, Mint. We could, IMEx here is the same thing as the intact database that we use. What I wanted to know was how representative of different species are these databases. I know a fair number of people here deal with plants, right? Now, I appreciate that Arabidopsis thaliana, shown here in the dark blue, is not the best model of all plants. I'm just perfectly willing to stipulate that offhand. But maybe we could throw in rice as well. We could chuck that in there. I didn't do this plot. What I asked was, how many interactions can I find for Arabidopsis thaliana? How many can I find for C. elegans? How many can I find for Drosophila melanogaster? How many for Homo sapiens? Musculus, the mouse, and S. cerevisiae. Right? These are all pretty widely studied organisms. So having counted up how many interactions I could find for each of those species in each of these databases, I asked, how do those match to each other proportionally? So something like Gene Ontology or KEGG is pretty good, frankly. We see a pretty wide distribution of species information. It's not like these are the human-only show or the mouse-only show. These are giving us information about all of the species to pretty good proportion. When you get to Wiki Pathways, though, or intact/IMEx, look at that huge yellow bar. Homo sapiens is just stealing the lunch of everybody else. So if you're working in clinical protein-protein interactions, those databases are brilliant. If you're working in plants, I can't say they have a lot for you. Now this is just 6 of the 702 pathway and protein-protein interaction databases out there. If you have an organism of interest that really your lab focuses on to the exclusion of others, I would suggest you go to the website and just enter it and see which databases say they support it. So what we can see is that at present, if you're working in human, pretty much any of these databases would be fine. But if you're working in something that's a plant, it seems pretty clear that services like IMEx/intact and Wiki Pathways may have less interest for you. Lots of takeaways. I know that was kind of a day at the zoo. Lots of different technologies all at once. Learning a transcript sequence gives you a lot of power in using sequence homology. You have a lot of different resources that you can go to with that. Expression data, if it's available in your species, has probably already done quite a lot of work to help you know gene-gene networks within that. And even if your species doesn't have a bunch of microarray data available for it, through homology, through orthology specifically, it's probably possible to discern which of your genes are to interact like they would in another species. Colocalization and protein-protein interactions are definitely a pretty heavily experimental place to be. If your focus is on what interactions does this individual gene product have, I think that there's a lot of room for you, for example, in co-immunoprecipitation. That's a really easy strategy to apply, and there are lots of labs here ready to help with stuff like that. However, it's still very much true that pathway and network information skew rather heavily by organism. It is worth taking some time to discern whether this service has entries that are of value to you before you just plunge in and assume, of course my species is covered. It may not be. And that is a wrap. Thank you.